Welcome to another lecture by MedicoMedics. Physiology Chapter 5, Resting Membrane Potential and Action Potentials. So in this lecture, we will talk about what is membrane potential? What's the concept of the resting membrane potential, RMP? What is action potentials, AP? Talk about phases, propagations of action potentials, its importance, mention some differences between RMP and AP between the cells. So is the resting membrane potential, for example, the same between a cardiac, heart cell, and a neuron, nerve cell? We will also mention some disorders related to membrane potential and end with a summary. Now, membrane potential refers to the electrical voltage difference across a cell's plasma membrane, and it arises due to uneven distribution of ions between the intracellular and the extracellular environment. So what is a membrane potential? We can define it as the electrical difference between inside and outside of a cell. Now, this potential plays a critical role in cellular functions, mainly in excitable cells like muscle fibers and neurons. Now, typically on the inside of the cell, you will find negative charge, and this is primarily maintained by large anionic proteins, like, for example, finding a lot of potassium, and also the selective permeability of the membrane to various ions. And contrary to that, on the outside of the cell, we will find positive charge. And why the extracellular environment typically has a positive charge is due to the higher concentration of sodium ions and chloride ions. Now, another important concept is resting membrane potential. Now, we measure, so the magnitude of the membrane potential is measured in millivolts, and it represents the electrical difference across the membrane. Now, a typical resting membrane potential in most animal cells, including in neurons, is approximately minus 70 millivolts. So this means the inside of the cell is about 70 millivolts more negative than the outside when the cell is at rest. And this resting state is maintained by ion channels and pumps, which regulate the movement of charged particles. Now, what creates this charge difference? So let's go and discuss a bit more in detail the ions and the mechanisms responsible for it. Now, as we mentioned, the resting membrane potential is the electrical charge difference across the cell membrane when the cell is at rest. And what does rest mean? It means that it's not actively sending any signals. Now, it arises due to the uneven distribution of ions, mainly sodium, potassium, and chloride across the membrane and the selective permeability of ion channels. And let's take a look at ion distribution. So we have sodium is high on the outside, low on the inside. Potassium, high on the inside and low on the outside. And chloride, similar to sodium, is high on the outside and low on the inside. So the inside of this cell is negatively charged compared to the outside with a potential difference, like we mentioned before, of about minus 70 millivolts in, for example, most neurons. Now, what is it that's maintaining this state, right? This state or this membrane potential is maintained primarily by sodium-potassium pump, which actively pumps out 3 sodiums or transports 3 sodiums out of the cell and transports 2 potassium into the cell. This aids in creating and maintaining concentration gradients. But we have another player that's also involved, and that's these leak channels we have, especially potassium channels. These allow ions to move down their gradients. So potassium leaks out more than sodium leaks in, leaving the inside more negative. So just to make it simple, imagine sodium is on the outside and potassium is on the inside. Now, if there are channels, right, transporters, whereby sodium can enter and where potassium can leave. Now, if there is a balance, let's say, 2 to 2, then there would be no difference between the two. But we mentioned that the sodium-potassium pump pumps 3 sodium out. So if we have sodium inside of the cell, 3 of them leave when 2 potassiums enter. So more, you can think of it like more positivity is leaving the cell. Therefore, we have a state of negativity on the inside of the cell. So that makes sense. But another way to maintain more negativity inside of the cell, as just mentioned, are these leak channels, whereby, let's say there is a 1-to-1 between sodium and potassium every minute, but every 30 seconds, potassium also gets out of the cell through these leak channels. What would then happen? Let's say we already have minus 50 millivolt, and now potassium leaves as well. So that would get it closer to 0 or the reverse. Well, we're losing something with a positive charge, so we're going to get further negative. So all of this aids in keeping the inside of the cell more negative. Now, why is all of this crucial? Because it sets up the cell for electrical signaling. So the resting membrane potential is crucial for excitable cells like neurons and muscles, because it sets the stage for action potentials and signal transmission. Because if you remember, we said this is the resting membrane potential, and rest meant that it's not actively sending signals. So this is the preparation stage, let's say, for what is to come, which is action potentials. You can also, let's clear this up, see here in this illustration that on the outside of the cell, we have a bunch of sodium, and on the inside, we will find more potassium. Now we have the sodium-potassium pump here, whereby sodium goes out and potassium comes in. But also an illustration of potassium leak channels, right? As you can see here, getting out. Now, once the resting potential has been established, how do cells use it to send signals? Let's now move to explore action potentials. Now then, what is an action potential? So it is defined as a rapid, temporary change in membrane potential that allows for communication. Essentially, the signal that is supposed to go off and go send a message somewhere. So you can think about action potential as being like a text message. It's how cells send information quickly. And it occurs in excitable cells like neurons and muscle cells. So what's the purpose here? Well, for neurons, it's to transmit signals along long distances. For muscles, it's about triggering contraction. Now, let's talk about the phases of an action potential. So we talked about the resting membrane potential. Here it's called the resting phase, or the resting state, to be more precise. So here the membrane is at minus 70 millivolts. And sodium-potassium channels are closed. Now, you may wonder, as I did in med school, a bit of a philosophical question, which is, why is it closed? I thought this channel was constantly open, pushing potassium in and sodium out. Now here's a brief attempt at an explanation for this, meaning why are the channels closed at minus 70 millivolts? Well, one reason is we have something called voltage-gated ion channels, and these are very sensitive to membrane potential. So these are channels, these voltage-gated ion channels, only open at specific voltage thresholds. So we are at minus 70 right now, right? And we can move towards 0. Let's say at minus 50, a channel could actually open up. And that would have consequences. Something comes in, something goes out, the charge would differ. Let's say at minus 30, some other channels open, having other consequences. So this is what we mean by voltage-gated ion channels. Now, at rest, if we're talking about, let's say, a neuron, at rest, the neuron is not stimulated. So these channels, therefore, will remain closed. And we just wait at resting membrane potential, waiting for some activity to happen. And the idea is to maintain this resting membrane potential, because imagine if these channels were actually opened. The ions would freely move across the membrane, disrupting this resting potential. And so instead, we have these sodium-potassium pumps, which actively maintain the stable environment, right? Making sure that we have this negativity. But the question was, why is it then closing at resting state? Because we want to prevent uncontrolled depolarization. So if the sodium channels just stayed open, sodium ions would continue to rush in, depolarizing the neuron. If potassium channels were open, potassium would leak out, making the neuron hyperpolarized instead. So the controlled opening of these channels during an action potential allows for proper nerve signal transmission. We also have selective permeability at rest. So the membrane is more permeable to potassium due to these leaky potassium channels that we talked about. And that allows for potassium to sort of diffuse out. However, it's not very permeable to sodium, so sodium cannot easily enter the cell the same way. And it is precisely this imbalance that creates a stable negative resting potential at around minus 70. So in short, the ion channels stay closed at rest to keep the neuron in a ready-to-fire state, while maintaining the necessary electrical gradient for action potentials. So I hope that clarified a little bit if you were also thinking, why are these closed? How come we kind of stop at minus 70 and why isn't it just continuously becoming more negative or positive? Because the whole idea is to keep the neuron, the cell, ready to fire when needed. And for that, we need to maintain the necessary electrical gradient. Okay? Now that's been clarified. Let's move on to what happens after that. So we're at resting membrane, minus 70. Sodium-potassium channels are closed. Then we have activity. Now we have depolarization. So what happens is that sodium channels open and sodium rushes in. Here you can see sodium from the outside entering. So what happens as a consequence of that? We had minus 70, right, on the inside. Minus 70 millivolts. If sodium enters and rushes in, these positive ions are entering into this negative area. What happens? Does minus 70 move more negatively towards minus 90 or opposite to that towards, let's say, 0? Well, you're adding 5 plus, let's say. to minus 70. That would give us minus 65, which is closer to 0, right? So positivity enters into the negative space, making it less negative. So the membrane potential becomes indeed less negative. And that allows for us to move towards other thresholds. Remember, as explained here, at minus 50, for example, some channels might open. At minus 30, something else might open. And then you reach plus 20, and let's say that's the threshold for the signal to send its message. We're just making up numbers here, but this is the key idea. We want to reach this number for this message to go. So when we say the cell has depolarized, it has reached this level, the signal has gone. Now what happens? Now we go into the process of repolarization. So we want to sort of get back here, ready for another firing. So what happens is that sodium channels now close and potassium channels open. This makes potassium being able to leave the cell, as seen here. And this will help us in restoring the negative potential again. We also have hyperpolarization, where potassium channels remain open a bit longer, causing a brief overshoot. So we get a bit more negativity, minus 80. And so during this, we restore the resting potential through the sodium-potassium pump, mainly. So think about it, because it can get tricky figuring all of this out. But if you understand it one time properly, that, let's clear it up, that we are at some resting state value, x, we want to get to y, right? So we need to get from minus x to, let's say, plus x, plus y. At plus y, something gets fired. The message gets sent. And there are a lot of sub-steps, steps in between here. And at this step, one channel opens, and another step, another channel opens, and here something else happens. Once we have reached y, and the signal has been sent, what should happen? Well, we need to get back to minus x. So understanding the stepwise elevation towards y, you can then work your way backwards to get back to the resting membrane potential. Now, let's ask the question, how does the action potential propagate along a neuron? Let's find out. Now, before we continue on with the propagation of action potentials in a neuron, we're just going to make a little detour and repeat something, because it is commonly an area where a lot of students might have a difficulty grasping the concept. So the question is rather simple. Why does potassium leaving the cell make it more negative? Because potassium is positively charged, as you can see by that plus sign. Now, inside the cell, at resting membrane potential, meaning at minus 70 millivolt, the concentration is higher inside than outside. We mentioned that. So when this potassium then leaves the cell through potassium channels, positive charge is lost from the inside. So you need to be able to conceptualize two things simultaneously. One, when they say potassium leaves the cell, what is actually happening is something with a positive charge called potassium is leaving the cell. And since fewer than positive ions remain inside the cell becomes what? More negative. Now, does the plus or minus signs indicate charge? Yes, that's exactly what it means. Plus signifies that this ion is positively charged, and minus that it's negatively charged. For example, sodium has a plus, So it's positive. Potassium plus, positive. Chloride, negative. And calcium has 2 plus, so it has a double positive charge. So I hope that makes sense. Now, what happens if chloride with a negative charge enters the cell? Well, chloride is negative, so if it enters, it will add a negative charge to the inside, which then makes the inside of the cell even more negative, so it becomes hyperpolarized. Now, neurons use this to inhibit action potentials, making it harder for the neuron to fire. Because remember, we needed to get to, for example, plus 30 millivolt for the action potential to go off. If we have negative charges entering a already negative environment, we will continue to make this cell negative to, let's say, 80 millivolts. So the distance, the gap between it now reaching its threshold has increased. So what does all of this mean for neurons? Well, depolarization occurs when there is more positive charges, meaning sodium has entered, making the inside of the cell less negative. Repolarization, it's returning to the negative state. So potassium leaves, making the inside more negative. And hyperpolarization, it's extra negative. So, for example, chloride with negative charges enter, or potassium keeps leaving. So we hope this little detour helped clarify some concepts. Now then, back to propagation of action potentials. So the main point here is that action potentials travel along the axon in one direction. And this here is an illustration of the axon. And let's briefly orientate ourselves with what we're looking at here. So we have the cell body of the neuron indicated in this section here. And it contains the nucleus and where information is processed. And we also have these dendrites sticking out. And these are branch-like structures that receive signals from other neurons. Then we have the axon. And this is the long extension that transmits signals away from the cell body. So in this direction. We have something called myelin sheath. This is a fatty, as you can see here illustrated in this yellowish color. It is a fatty layer that speeds up signal transmission. We have nodes of Ranvier. These are gaps in between. Let's clean this up. There we go. So the nodes of Ranvier are these gaps in myelin that allows fast signal jumping. Then we have the axon terminals, and these release neurotransmitters to communicate then with other neurons. So propagation works like this. Essentially, the action potential is traveling along the axon in one direction. The mechanism here is depolarization at one segment triggers the next segment. So it's like dominoes. Previous segments then enter into a refractory period, so it becomes temporarily inactive. We mentioned an important structure, the myelin sheath. These speed up conduction by allowing the signal to jump between the nodes of Ranvier. So example, single transmission from the brain to a muscle. Now, to make it a bit more clinically relevant to you, so you have a concept of action potentials, the neuron, the propagation of these potentials, and now you can hopefully start visualizing that, okay, inside of a cell, it is ions are entering, exiting, we're getting closer to a action threshold. Once we reach the threshold, a signal is sent off, and it travels in the neuron in the way we have described it here. Now, we mentioned, for example, so let's make this clinically interesting. We mentioned this structure called myelin sheath here. And these were the fatty layer that helps speeding up signal transmission. So in more detail, we could add that the myelin sheath, aside from being a fatty layer, is indeed a protective structure that wraps around the axons of the nerve cells. And what it does is it insulates them and therefore accelerates the transmission of these electrical signals. And it is produced by something called Schwann cells. And these Schwann cells in the peripheral nervous system, it's occurring in the peripheral nervous system and by oligodendrocytes in the central nervous system. What is typically high yield is to remember Schwann cells. These are what gives rise to these myelin sheath. Now, why is this interesting clinically? Because there is a quite famous disease, perhaps you've heard of it, that is associated with the destruction of these myelin sheath. And that disease is called multiple sclerosis, or shortened MS. And in multiple sclerosis, what is happening, I'm just giving a brief overview here, but what is happening is that the immune system, our immune system, is mistakenly attacking this myelin sheath. And that therefore then leads to what? Well, if we understand what the function of the myelin sheath is, what will happen then in MS by the destruction or attack on this myelin is that it will lead to slowed or disrupted nerve signal transmission. And if signal transmission is slowed down or damaged or disrupted, you can imagine whatever this neuron is sending signals to, let's say to a muscle or to our vision or systems involved in coordination, whatever it may be, will be impacted. So we may see muscle weakness, we may see vision loss, we may see impaired coordination, etc. Now we have, and you will indeed study dedicated lectures on multiple sclerosis, for example. But the idea here is to connect what is clinical, what can present in your office one day. Let's say the patient with a vision loss in one eye and muscle weakness, etc. And you then find out that, oh, there is an immune system involvement where there is a destruction of these myelin sheath. And how do you then understand this symptom picture? Is because you have now understood that, oh, the thing it's destroying is the ability of transmission. And transmission is directly connected to, or the other side of the coin, is the concept of how action potentials or signals indeed are fired. And this action potential is directly correlated to concepts like resting membrane potential. And this is based on the movement of ions and cations, positive, negative charges, and what's happening inside of the cell. Transporters on the cell membranes allowing for things to go in and out, changing the charge. So hopefully, thus far, you have now an overview idea of something that enters literally the symptoms. You see them in your office and how they're related to this very basic and fundamental part of your medical studies. Now, obviously, each of these steps can be dealt with in detail. Just a symptom and clinical picture is a subject you need to study as you go along. But also, immunology was involved here, like there is an autoimmune problem. So your concept and understanding of immunology is a subject you will continuously study, and so on and so forth. So what is indeed the importance of action potentials? Essential for nerve signal transmission, like reflexes, sensory input, muscle contraction, like heartbeats, movement, etc. Disruptions, neurological disorders, like we just mentioned, multiple sclerosis. And it is precisely due to demyelination. If you remember, myelin sheath. And the destruction or disruption of this is typically called demyelination, the loss of it. And of course, disruptions can lead to cardiac arrhythmias, so abnormal heartbeats. So let's just mention some key differences here. Because here is another interesting thing that may confuse a lot of students or take a lot of time for students to make sense of. So we ask the question here because we make learning easy. So the question is the action potential structures mentioned in the previous slides the same for cardiac cells and all other cells? Because we focused on the neurons, right? And the answer is no, it's not. The action potential structure differs between neurons, cardiac cells, and other excitable cells. However, the basic principles, so depolarization, repolarization, etc., these are similar. But the ion channels, the duration, and the phases will vary depending on the cell type. So let's take two for example. We have neurons here, okay? Let's look at neurons. Now, it has a duration of 1 to 2 milliseconds, so it's really fast. It has a resting membrane potential of minus 70 millivolts. It depolarizes, and the main ion involved in that is sodium. The influx of sodium into the cell. The repolarization is primarily connected to potassium efflux, so potassium leaving the cell. Does it have spontaneous firing? No. Does it have a plateau phase? No. And its fast conduction, as we just mentioned, is due to myelination. Right? These myelin sheath, for example, and the nodes of Ranvier, etc. Now, what about cardiac myocytes or cardiac muscle cells? These have a duration of 200 to 400 milliseconds. So it is much longer than neurons, right? Furthermore, some similarities, it has the same depolarizing ion and repolarizing ion. So it's going to be sodium influx and potassium efflux. Does it have spontaneous firing? No. But it has a plateau phase. And its resting membrane potential is different. And one of the main reasons for why it has a plateau phase, which is unique to it, right, is because this helps it maintaining contraction for effective pumping, right? We're talking about the heart now. Unlike neurons, which we want firing fast and resetting quickly. Now, I said we were just going to compare two, but let's add one more, which are pacemaker cells. These are cardiac pacemaker cells. And you will learn about all of these when you study the physiology of the heart, the electrical system of the heart, so to speak. Here we have a duration of 150 to 250 milliseconds. And the depolarizing ion here is calcium, actually. The resting membrane potential is unstable, meaning these cells spontaneously depolarize, setting the heart rate. So, in summary then, neurons fire quick action potentials. Cardiac cells have longer action potentials to ensure proper heart contraction. And we have fast conduction in neurons due to myelination. And in pacemaker cells, here we find something unique where we have spontaneous depolarization to regulate heartbeats. So hope that clarifies some questions that one may have. Let's go on. So some disorders related to membrane potential, like we mentioned again. Multiple sclerosis, so damaged myelin slows or blocks signal propagation. Epilepsy, where we have overactive neuronal firing due to disrupted ion channel function. So now the issue is at the channel. Here we had myelin, meaning the signal propagation was the issue. We can have hyperkalemia, which alters resting membrane potential, leading to muscle weakness or arrhythmias because we have too much potassium. Of course, if you think about it, we made the connection between, for example, MS or whatever pathology, right? And we connected it to the signal transmission. We connected that to the world of resting membrane potentials and action potentials and concepts like threshold, etc., etc. Repolarization, depolarization, and that this was all based on the influx and efflux of ions. Now, imagine you have too much potassium. Imagine you have too little sodium, too much chloride, too much calcium, too little calcium. And we have names for all of these. Actual diagnoses like hyperkalemia, hyperkalemia, hypokalemia, hypernatremia, etc., etc. So these are not just made up for the fun of it. It's indicating directly because it has consequences. You will learn about it as you go on in your medical studies. That if you have too much potassium, sure, there will be consequences here, and that will have consequences here, etc. And then you see that in your office. So in summary then, resting membrane potential is the foundation for electrical signaling. Action potentials allow rapid communications in neurons and muscles, and disorders arise when ion gradients or signaling pathways are disrupted. And that brings us to the end of this lecture. Hope you enjoyed it. Continue now to the next chapter.